Hi, my name is Frederik Leerjaert, and in this talk I would like to highlight the importance of collections in taxonomy and also talk about the future of phycological collections, and in particular how digitization will be important to facilitate taxonomic research in the future. One thing that is striking when looking at herbarium collections is that not much seems to have changed over the past 200 years or so. A herbarium specimen collected in the 19th century still looks very much like a specimen collected today. And also methods for collecting and preserving herbarium specimens haven't changed very much. And we still press and dry seeds on paper, and these sheets can be kept for centuries under the right storage conditions. So herbaria have been this kind of open data sources before open data really existed, and over these many years herbaria have served many scientific roles, in particular for taxonomy. Globally there are more than 390 million plant specimens stored in more than 4,000 herbaria. It is difficult to find exact figures on the number of phycological specimens, but there are many for sure. Thousands of herbarium specimens of algae, microscope slides and other types of collections are housed in hundreds of institutions worldwide. Some of these phycological collections are well known, but many other collections remain largely obscure. So one question that arises is, are herbaria still relevant today? In other words, is it still useful to collect specimens and keep vouchers? My answer, of course, will be yes. We see that herbarium specimens are still widely used today. This is illustrated here by this publication of Hibberling et al. Based on computational text analysis of more than 30,000 studies in plant science. And their analysis showed this exponential use of herbarium specimens. Maybe not entirely surprising, given that also the number of publications is rising in the same way. The study was based on plant sciences, but also included algal, floristic and taxonomic studies, indicated here by this red arrow. And so, although most phycologists and taxonomists in particular acknowledge the value of collections and herbarium specimens, I'd like to briefly reappraise the herbarium specimen here. When we look closer at a specimen, we can see that each specimen really has its own story to tell. So a single herbarium sheet contains various data. The label tells you where, when, and by whom, and in which type of habitat the specimen was collected. It also contains generally a species identification. And from the specimen itself, morphological and anatomical data can be extracted, including trait data such as thallus height and front area, for example. But also genetic data and biochemical data can be extracted, possibly also species interaction data, for example, if the specimen was found epiphytically on another specimen. So jointly, all these data represents various evidence for biodiversity. Taxonomy and systematics have always been at the core of use of herbarium collections, and we see that their use has been relatively constant over time, as illustrated in this left graph here. In taxonomy, herbaria have served two main purposes. In the first place, they are places for storing vouchers, including types, and this ensures repeatability of the taxonomic research. In the second place, herbaria are also places for species discovery. We know that a large amount of undescribed species are sitting in herbaria waiting to be discovered. But we also see the emergence of new uses. An important one are DNA analysis from herbarium specimens, and these have been important in DNA taxonomy, for example, but also for phylogenetic reconstruction in biogeographical studies. But we also see that other new uses of specimens are emerging. This is illustrated here in this right graph. For example, in plant sciences, functional traits, including leaf area, stomatal density, seed mass, and so on, are being extracted from herbarium specimens across time to study responses to climate change, for example. So we are seeing this renewed interest in herbarium specimens with new uses that go beyond what the original collectors could have anticipated. And also in the phycological literature, we see that new uses of collections are emerging, ranging from phylogenetic and biogeographic analysis, here are just a few examples, to the tracing of invasive species, for example. But also other uses, for example, herbarium collections have been used to infer changes in seaweed communities in response to climate change, although there has been some criticism on the use of collections for these purposes, for example, because they are often not representative to study these long-term changes. But anyway, an emerging new uses of herbarium collections also in phycological studies. Although we see that collections are still widely used today in phycology, one traditional problem with herbaria is that they are sometimes difficult to access. We have to dig into the literature to find out where specimens are housed. And to study these collections, they have to be sent on loan, or we have to travel to visit the institutions. And in some cases, collections are physically inaccessible for different reasons. So to overcome these problems, we have seen in the past decades many initiatives from institutions worldwide to start digitizing their collections and make them openly accessible online. And this is giving an unprecedented access to herbarium collections to the research community and to the taxonomic community in particular. I've just listed a couple of collection portals here, some of which are specifically focused on seaweeds, including the American macroalgal herbarium portal and the UK platform Seaweed Collections Online. Just to give our own institution as an example, Mesa Botanic Garden started digitizing its collection, about 4 million plant specimens, 6 years ago, and we are about halfway today. We use in-house equipment as well as high-speed conveyor belt imaging technology in collaboration with a private company, Picture. And this allows for digitization of thousands of specimens per day at relatively low cost. We use standardized workflows to get these specimen images online, along with the data on our herbarium portal, as well as on international open access platforms such as GBIF. And we are currently digitizing also our seaweed herbarium, which we recently acquired from Ghent University, and we hope to have the entire collection out soon. So we see that collection data is gradually becoming available online, which is great. But in general, the landscape of natural history collections is still a very fragmented one. Collections are kept in thousands of different institutions worldwide. These include large institutional collections, but also smaller lab collections in universities, for example. And in these many institutions, collection data are stored in separate databases, often using different data standards. And for many collections, we don't really know what's in them, so these collections remain largely hidden. So there is still a strong need to try to unify these collections to make them better accessible. And defragmentation of collections cannot be done at the physical level, so it has to be done at the digital level. And there are a number of large infrastructure initiatives that aim to digitally unify the collections. And these include the American IDBio, the National Specimen Information Infrastructure of China, Atlas of Living Australia, and the European Distributed System of Scientific Collections, or DISCO in short. So DISCO aims to unify all European natural history assets into a single digital collection, which will greatly facilitate taxonomic research, as well as enabling new research by interconnecting different collections and other data streams. And currently 120 institutions from 21 countries are participating, in DISCO, dealing with a total estimate of about 1.5 billion specimens. So the whole idea of DISCO and other initiatives alike is to digitally unlock the collections by creating digital specimens, which we can interact with in a whole different way than we could with physical specimens. So in this digital or extended specimen concept, the physical specimen remains at the core, but is now extended by a digital twin, which contains information on the collection event, the identity, and a high-resolution image. And using information technology, it is now possible to link this digital specimen with other digital specimens and associated data. For example, we can link a holotype with its isotypes, other specimens. We can link other objects such as microscopic slides or silica material. We can link the specimen with other information such as field photographs, literature, trait data, DNA sequence data, and so on. So this kind of data integration will have a significant impact on taxonomy. It will become easier to find specimens, and it will become easier to link those specimens with taxonomic and nomenclatural information. And what is important in this new digital herbarium landscape is that we use standardized identifiers and data to improve data integration. Unique barcodes for physical specimens is not a new thing and have been used by institutions for many decades, but these can now be translated into these unique persistent identifiers that identifies the digital specimen persistently and globally. And resolving this identifier will always return the digital representative of the physical specimen illustrated here. And in turn, this unique identifier can be used to unambiguously link that specimen to other data sources, to literature, classification, field photographs, species profiles such as traits, geographical ranges, and DNA sequences. Linking specimens to DNA sequences is especially important because we know that a very large proportion of sequences in GenBank are either not identified to the species level or species identification is doubtful. So linking sequences to specimens through these unique persistence identifiers is often the only way to resolve their identity. And so the role of data integration is very important as we enter this digital herbarium landscape. And one example is GBIF, which is one of the main aggregators of biodiversity data information, including collections, but also other sources of data, such as literature and genetic data. So we're not quite there yet. Biodiversity informatics is a growing field, but I think these are very exciting times for phycological collections and for algal taxonomy. So to conclude, seaweed herbarium collections are still relevant today, and their uses are expanding. We must keep curating herbarium collections, both physical and digital, and the digitization of collections and integration of data will have a significant impact on algal taxonomy. And with that, I would like to thank you for your attention.